Good evening, everyone. Thank you so much for subscriptions, for likes, for comments. It makes us improve each day. After discussing degradation of amino acids and classification based on the degradation products, I thought it wise to discuss the urea cycle. When you look at the urea cycle, you ask yourselves, where do the amine group go? We have the amino terminal end of the amino acid. We also have the presence of the amine group on the R side chain. Where does that amino group go? Because if it's left in the system, it becomes toxic. And that is how important the urea cycle is. Because the digestion of those proteins will release the ammonia, or that nitrogen component. The degradation of amino acids will release it as well. So the urea cycle has to come handy and help the situation before it gets worse. So that's how amino acid structure is: carboxyl group, amino group, the R side chain, the hydrogen, and the alpha carbon. So the amino group, which you can see there, can go to waste or can be reused. So that is what we are going to look at, because we are interested much on where the NH3+ or the NH2 go. So ammonia originates from degradation of amino acids. Basically, even degradation of proteins or digestion of those proteins also generate ammonia. So they can be from the digestive enzymes, proteins released by digestion, muscle proteins, hemoglobin, intracellular proteins, different proteins can generate the ammonia. But if it's left in the system, this ammonia, which is generated by degradation of those amino acids and digestion of those proteins, is toxic, especially to the central nervous system, because it reacts with alpha ketoglutarate. Therefore, it will make it limiting for the TCA to get enough of alpha Kg, and this can decrease the ATP level. So even the liver damage and some of the metabolic disorders have been associated with high levels of ammonia, and it has led to tremor, problem in speech, people have slurred speech, problem in vision, people have blurred vision, which can lead to coma and death. So we have to find a way of eliminating the ammonia. But then the normal concentration of ammonia in blood is always 30 to 16 micromolar. So when it is higher than that normal range, then definitely there is a problem. So we have to find ways of eliminating the ammonia. So we saw the two options of where this ammonia go. It can be reused, or it can go to the urea cycle. When it is reused, it is used much in the synthesis of amino acids. For example, aspartate, just by adding the amine group, will form asparagine. Glutamate, by adding an amine group, will form the glutamine. Also, the ammonia can be used in the nucleotide synthesis as well as synthesis of some biological amines. So the second choice is it has to go to the urea cycle, where it is going to be excreted as a nitrogen product. So it shows the options of the ammonia itself. So in the urea cycle, it will form an intermediate of the TCA cycle, fumarate. Then the fumarate can go to the TCA cycle and then it is going to generate for us the reducing equivalence. So it can be reused or it can go as a waste. Before urea cycle is done, there has to be we need these 5 steps for us to dive into the urea cycle. So the amino group has to be removed at the first step. Then the second step is you take the amino group to the liver for nitrogen extraction. Then the third step, entry into mitochondria. The amino group, will enter into the mitochondria, and then you'll prepare the nitrogen to enter the urea cycle. Then the last step is the main pathway of the urea cycle. During our study in Form 3, I think, we were looking at excretory forms of nitrogen, and what I could remember was uricotelic animals, and that one I could remember the birds. The product, the nitrogen product in birds was uric acid, and now even the reptiles. And what I could also remember much were the aquatic. The aquatic, they were known as aminotelic. So ammonia. And then we have urea, terrestrial vertebrates. So just like us, urea, and also sharks. So the amphibians and the fishes, aquatic and aquatic vertebrates will always release it as ammonia as a nitrogen form. Then the terrestrial, the one on land, and even sharks will release it in form of urea. Then the birds and reptiles will release the nitrogen as uric acid. So those are the different classes on how nitrogen is released as per different organisms. So for the nitrogen to be released from the amino acid, we saw the different steps. So there has to be a carrier, nitrogen carrier. So the nitrogen carriers, we have glutamate, glutamine, and alanine. Glutamate will transfer the amino group within the cells, and then the glutamine will transfer 2 of the amino acids between the cells, and then lastly we have alanine, which will transfer the amino group from the tissues, that is the muscle, into the liver. So those are the 3 nitrogen carriers which are known. And then lastly we will have the urea cycle, where now the ammonia will be released via the urea cycle. So if you talk about the urea cycle, you can see there it has 5 steps, 5 steps only. But at the first point you can see the ammonia is highlighted in red or pink. It has to combine with carbonic acid, and there has to be source of energy in form of ATP. The enzyme which catalyzes the formation of carbamoyl phosphate is known as the carbamoyl phosphate synthetase. So there will be a release of 2 diphosphates and a phosphate. Then the second step, ornithine will combine with carbamoyl phosphate to form acetylene. I told you these structures is just interference with only 1 carbon, not a whole lot, and then it becomes easy. You can see the carbamoyl phosphate is NH3C double bond O, a phosphate. Then we have the ornithine with the carboxyl, with the amines, 3 of the CH2s and NH3+. Then it is going to form citrulline. So citrulline is just the same structure as whatever we had here. In fact, the carbamoyl phosphate will come and add at the end of the structure of ornithine to form the citrulline. When you look at the structure of citrulline, just addition of the carbamoyl phosphate, the phosphate is released. You can see the P1I going here, not P1, Pi getting released. So you are forming the citrulline, just the same structure of ornithine. What you add there is only the it's only the carboxyl group and the amine. The phosphate has been removed. Then from that carbamoyl phosphate combine with ornithine to form citrulline. The third step is aspartate combining with citrulline to form argininosuccinate. The enzyme is argininosuccinate synthetase. After forming the argininosuccinate, then it has to be broken down. At that point where it is broken down, breaking down the liases have to break it down to fumarate and arginine. The arginine itself will proceed. The fumarate will connect to the TCA cycle to generate the reducing equivalence, NADH. It will combine at the point of malodehydrogenase and generate NADH. The arginine will go next step, where now the urea is going to be released. In that step, where the arginine is forming, it is going to release urea. Then the fourth step, that is where the urea is released. So that is how the nitrogens, the forms of nitrogen for those organisms which we've seen, based on the terrestrial, based on the aquatic, based on the birds, that is how the nitrogen is going to be released in terrestrial. That is the urea cycle. That is how the ammonia, which is generated from nitrogen, metabolism of those proteins, the degradation of those amino acid, it should not be toxic, it can be released through the urea cycle. A bit of the deficiencies which are involved in the urea cycle, it is just based much on which of those enzymes is defective. It leads to what condition? So the carbamoyl phosphate synthetase 1, because there is another carbamoyl phosphate synthetase 2, which is found in nucleotide metabolism. So the carbamoyl phosphate synthetase 1 deficiency, it is the one which is lead to formation of carbamoyl phosphate. So the carbamoyl phosphate synthetase 1. Formation of carbamoyl phosphate, because we have to form the carbamoyl phosphate so that it can link with ornithine and we generate citrulline. So the carbamoyl phosphate is generated because the amine group, the amino group, the nitrogen component, which is released from those terrestrial animals, it has to combine with carbonic acid and phosphate to form the carbamoyl phosphate, which is the entry point of the urea cycle, because we have to release it. It's toxic in the system. So if that enzyme in the formation of carbamoyl phosphate is deficient or is absent, then it leads to hyperammonemia. Ammonia will not be able to combine with carbonic acid to form carbamoyl phosphate. So you can see it's also lead to mental retardation and developmental delay. Ornithine transcarbamylase, the enzyme which was involved in formation of citrulline, if it is defective, you can see it is an X-linked disorder. So it is related with the sex chromosomes, X-linked. It is related with the sex chromosomes. It is not autosomal, it is X-linked. So we'll understand much during genetics, but by now we need to understand much the deficiency of these enzymes. How grave is it? Argininosuccinate synthase. That is a point where the citrulline condensing with aspartate, the importance of amino acids as well. It was leading to formation of arginosuccinate. So if that enzyme is defective, it means inability of citrulline to condense with aspartate. Then there'll be accumulation of citrulline in blood and excretion in urine. So these are two types of citrullinemia as a result of that enzyme being defective. Arginosuccinate lyase. Breakdown of that arginosuccinate to form fumarate and arginine. Arginosuccinate will be excreted large amount in urine, and the symptoms are very grave. So it is also classified as rare autosomal recessive disorder. And then the arginase, at the step where the rare is released, excretion of arginine in urine, it will accumulate and then it will be excreted in urine. Ammonia encephalopathy, so much of the ammonia concentration in blood and other biological tissues. And in this case, we're talking much about the alpha Kg is depleted from CNS. And that is where you can see it will inhibit the TCA cycle because we need it as intermediate.